So now we're going to talk about balance. And since you already are quite familiar with the hearing, believe me, the mechanics are pretty much the same. So we're going to start with the structure called vestibule, which is circled red in that image. So vestibule consists of two subparts, a utricle and a saccule. Utricle and saccule are responsible for testing or detecting linear accelerations. Okay? Whether it's a horizontal acceleration or vertical acceleration. Within saccule and within utricle, there are receptors called maculae. You can see the macula of the utricle here and the macula of vestibule here. So these spots are maculae. Now, the second part of the inner ear that is also responsible for sensing the balance are semicircular canals. So semicircular canals, there are three of them because we live in the 3D space. They are responsible for sensing rotation of the head, or so-called dynamic equilibrium. Okay? So first, let's talk about the saccule and the utricle. So in this schematic representation, the central part, the one that I just circled green, is the vestibule, and these two chambers, utricle and saccule, contain the receptors that are called maculae. They monitor the linear position of the head. They respond to linear acceleration. Okay? So each macula has pretty much the same structure. Let's take a look. Among the supporting cells are hair cells. Just the same hair cells as the hair cells in the ear. Does that make sense? The stereocilia, the hair, are embedded into this otolith membrane. You see, thick layer with little crystals of calcium carbonate that are called otoliths, which can be translated as the hair stones. So I can't stress enough that this membrane, the otolith membrane, is pretty heavy. Okay? Sounds good? Heavy. Well, I mean, for a small structure like that. Another thing that I want you to appreciate is that while the entire vestibule is the part of the bony labyrinth and is filled with perilymph, the utricle and the saccule are both parts of membranous labyrinth think I misspelled labyrinth. But, and filled with potassium-rich endolymph. Okay? You with me? We're gonna, no, it was to everybody, it wasn't. I know that you, yes, I do. Okay, so how are they activated? So look at this. Remember, your otolith membrane is real heavy. Okay? So imagine that your head starts moving into that direction. Right? So the hair cells start moving in that direction, but the otolith membrane, since it's very heavy and has huge inertia, will kind of stay in its original place for some time, which means this hair will get bent. Makes sense or not? The easiest way to think of it, like seriously, you are in the car and you press the gas pedal, like seriously press the gas pedal. You're blasting off, okay? What's going to happen to your head? You're going to go back, right? Because of the inertia. Because of the inertia of otolith membrane, when your head is moving, otolith membrane kind of stays behind and bends the stereocilia, which means, just like with hearing, stereocilia are bent. They allow potassium to get in, depolarize the hair cell, and release a neurotransmitter. Am I making sense? Now, if I personally, I myself don't know, like, are you moving forward, backwards? Basically, the idea is this. If you move into one direction, the hair cells will depolarize because the channels will open, and the increased frequency of impulses to the brain will be interpreted as, for example, moving forward. If you're moving backwards, because it's opposite direction, they're actually hyperpolarized. The frequency of impulses decreases and your brain interprets it as I'm moving backwards. Does it make sense, y'all? Now, here's a very interesting thing about that static equilibrium, folks. So have you been on like a really high-speed elevators? Yeah. That go like a lot of floors up. Like it takes a good minute to ride. Sure. So imagine you walk into that elevator and it starts moving. Can you feel it? When it accelerates, you can feel it. But then when it reaches a constant speed, you don't really know, right? Because there's no acceleration. Like in the car, like when you start accelerating, you're being pulled back. But when you reach the cruising speed, there's nothing. Yes, exactly, like an airplane, with no acceleration. Okay, make sense? So it only tests acceleration, not necessarily a movement itself. Now, going back to this, check this out. If the macula is located horizontally, like in the utricle, it will sense, well, movements that are in the horizontal plane, forward, backwards, left, right. Sounds good? In the saccule, the receptor, the macula, is vertical. So saccule is responsible for you feeling, the elevator starting going up or slowing down as it reaches your target floor. Make sense? Okay. Now what about semicircular canals? So within a canal, there is a semicircular duct. So in the duct, there is a receptor called quista ampullaris. So this whole thing is quista ampullaris. Now, if we enlarge that image, you see again that these are your old pals. The hair cell with all those stereocilia. But this time, stereocilia are embedded into the structure called ampullary cupola, which is very flexible and can bend the cupola itself. And I want to remind everybody that the semicircular canal is filled with endolymph. Again, potassium-rich fluid. So what's going to happen when you start rotating? The fluid, the endolymph, because you rotate, starts flowing, bending the quista ampullaris. And depending on which direction you are rotating, the frequency of action potentials can either increase or decrease. And your brain will interpret it as rotating left or rotating right. Does that make sense? What I want you to kind of think and make associations about, if you think about hearing, if you think about vestibule, the static equilibrium, the dynamic equilibrium, in every case, it's the bending of stereocilia and influx of potassium and calcium. In case of hearing, it's because the basilar membrane vibrates. In the case of static equilibrium, it's because otolith membrane is heavy and kind of bends, like moves the stereocilia backwards. In case of semicircular ducts, it's because endolymph, the fluid that fills up the semicircular duct, bends ampullary cupola and deflects the stereocilia. Okay? Now, in the PowerPoint, there is a YouTube video. It's kind of an interesting just to see what vestibular nystagmus is. So basically, if you turn someone, like if somebody would rotate me on the chair, my eyes will be jerking. That's called vestibular nystagmus. So pathways. What I want to focus you on, hear me out. Well, vestibular nuclei, sure. But the point is, my point, is that your vestibular receptors, they communicate to your cerebellum and to your muscles and joints. Why muscles and joints? Balance, yeah. Remember, vestibular apparatus, the stuff that we just discussed, does not maintain the balance. It tells your body, hey buddy, you are in, I don't know, you are rotating or whatever, right? You're moving forward, you're moving backwards, accelerating, this and that. And based on those inputs, your muscles respond through reflexes to maintain the balance. Remember, we're talking about reflexes, right? We all have the same reflexes, but in people with better balance, they sharpen. Just, they better at balancing. Okay? Sounds good? Reasonable? Awesome. Couple of pathologies. Motion sickness. So the exact mechanism is a no. But here's the idea. Mismatch of sensory inputs. So, imagine that you are driving your car. You're at the wheel. Are you looking on the road? I hope so. Your eyes, what do they tell you? Are you moving or not? You're moving. Your vestibular apparatus, what does it tell you? You're moving. Like, they are matching those inputs. Now, you sit in the back seat with a book or phone, and you're looking at your screen. From your eyes perspective, are you moving? No. From your vestibular apparatus, you are. Nobody gets sick when they drive. But if you're a passenger, especially when you're reading, you can get sick. Okay? Now, what happens in case of mismatch? For some reason, people still don't understand how, that mismatch creates the signal that stimulates the vomiting center in the medulla oblongata, and you have nausea. Okay? So, Dramamine, the anti-motion sickness drug, it does not prevent the mismatch, just blocks the vomiting center. Okay? And same story why people get seasick. Mostly because of the mismatch between visual and vestibular inputs. I happened to be in a relatively small boat on Lake Erie, and it was pretty rocky that day. We were fishing. Whenever I went into the cockpit and I didn't see the lake, I got seasick. The cure, I would just pop out, just look around. Okay, the boat's moving. The eyes tell me what's going on. Vestibular apparatus tells me the same. Symptoms subside. I can go back. It's absolutely normal. That's how people are built. Ménière syndrome, terrible condition. Your inner ear becomes filled with fluid, and that messes up the signaling. Do you understand what I'm trying to say? So, instead of sending you proper signals, it sends you signals that do not exist. Okay? And it creates a mismatch in a situation where there shouldn't be any. Right? You follow? Okay. Yeah, it's rare. So diuretics, basically trying to drain the fluid out of the inner ear. Complicated cases, the removal of the labyrinth. Questions? My favorite sense is the smell. Mostly because of the elegance of the mechanism, how we sense different smells. So we are going to start with the anatomy. Okay? We're going to go from sort of big to small anatomy. So this is the olfactory nerve, and this is the olfactory bulb. I want to point your attention at the fact that the nasal cavity and the brain are actually separated by a very thin layer of bone, which is a part of an ethmoid bone. It's called cribriform plate. I'm a bit paranoid about it. So, oh, for God's sakes. Okay. This. So it's fairly thin, right? All good? Now, when you start looking at your olfactory epithelium closer, here's what you're going to see. Hear me out. These are olfactory neurons. Okay? Good? When they bundle the axons, when they bundle, they form filaments of olfactory nerve. And if you look at this image, you may notice that those axons, those bundles, those filaments, they go through the bone, through the cribriform plate. So there are real holes. Does that make sense? It's going to become important in a minute. So these guys, and remember, this is which part of a neuron is that? Which part of a neuron is that? That's the direction of the signal. Axon. Which part of a neuron is that then? Yes, those are dendrites. They are called olfactory cilia. This is the only cilia in the human body that cannot move on its own. The cilia is completely covered in mucus. Why do you want mucus to cover cilia? Not only that, no. Smell. When you smell something, what is the actual stimulus? When, like, I have some substance like cloves there. You smell cloves. What is it getting in your nose? What? Can't hear you. Louder. Right, but what is it? Like, is it... Huh? Smaller. You on the right track. The M word. Molecules. Molecules. The smelly substances are molecules. These molecules have to be dissolved. And that mucus dissolves the odorants. So far, we good? Okay, we figured out why we need mucus. We figured out how the olfactory nerve is built. These are stem cells. Remember stem cells that can develop into something? Why do you want... So stem cells can develop into olfactory neurons. Why is this important? Can you kill the olfactory neurons? Yeah, you can kill them. When I was first year of college, now I can tell this, we had a Gen Chem lab, and we were producing chlorine gas. The same chlorine gas that killed a bunch of French troops in 1915. So instead of being a responsible chemist and, waving the gas at my nose, I had really stuffy nose. I had a cold. So I just stuck the tube into my nostril and took a good, proper sniff. Dude, no, I'm not that weak. I didn't pass out. I lost smell sensation for a week because I wiped out my olfactory epithelium, but I also cured my runny nose completely. It was great. So, stem cells replace it. Okay? So far, we making sense? So, the holes. Why is this clinically important? Well, first of all, because of drugs. The reason why people that use some recreational drugs are used by sniffing them, glue, sniffing glue. It's not about the smell, it's about the active compounds going straight up here, through the holes. Does that make sense? So it's a direct delivery in the brain. Second thing, have you heard about brain-eating amoeba? One of my favorite creatures. When people swim around, mostly kids, and snort the water when they swim, like, that's going to happen. the amoeba can be snorted in the brain, and it has nothing else to do as to start just destroying your brain. Causes inflammation, usually people die. Okay? That makes sense? Remember, amoeba doesn't want to be there. Now, why do we use the term like sniffing glue? People don't just, like move the air towards their nose. They sniff because when we sniff, we force the air towards the epithelium. We do sniff something if the smell is kind of faint, but when people sniff glue, they direct those active chemicals, active compounds, straight towards the cribriform plate, and that acts on their brain. Okay? So, this is the Nobel Prize for 2004. Okay? It's I think it's one of the most deserved Nobel Prizes ever. Linda Buck and some other dude, they shared the prize. So, think about this. Humans can distinguish about 10,000 different smells, which is remarkable. We're not the best, but it's good. Each smell is an individual molecule. We may have 10,000 receptors, each of them being a protein. The problem with that idea is that we have total about 25,000 genes. It's unlikely to sacrifice half of available genes just for a pretty lousy sense of smell. So the idea that Linda Buck and whatever, I can't remember the name of the guy, came up with is that it's a combination. And they called it an accordion theory. So, we have several sensory smell receptors, S. Okay, so let's take a look at S18. There is no particular reason why I chose it. So S18 can be stimulated by all those compounds. Okay, can be stimulated by heptanoic, octanoic, nonanoic acid, octanol, nonanol, whatever, doesn't matter. Now, what I want you to appreciate, check this out. It is most sensitive to nonanoic acid. So not only it can be stimulated by different compounds, but the strength of the stimulus is different for different compounds. Does that idea make sense? So a single receptor can be triggered by several compounds, and these compounds can create a signal of different strength. If you look at any individual compound in this list, say octanol, it triggers 4 different receptors. Okay? Now, this is why they called it an accordion theory. You're probably familiar with a music instrument, accordion, because when a chemical strikes the receptors, the unique combination of the receptors that send the signal to the brain, in case of octanol, it's S18, S19, S49, and S51. It's like 4 different notes, 4 different keys on a keyboard that create a unique chord of stimuli. Does it make sense? So at the end of the day, we have about 400 sensory smell receptors. And technically speaking, when we say like 10,000 different smells, it's pretty much unlimited. Because if we create a new molecule that strikes a new combination of receptors, it's a new smell. So, to summarize, each receptor senses several different odors. Each odor can stimulate several different receptors. Like, look at this example here. So imagine you have an order like this. It's a schematic representation, right? This guy. So this smelly molecule will stimulate this in these receptors. This smelly molecule will stimulate this, and these receptors. Am I clear? So at the end of the day, we can sense a lot of smells. What's really important, each neuron, each individual olfactory neuron carries only one type of receptors, which makes sense because if a neuron can be triggered by different orders, it's going to be cacophony. Do you understand what I'm saying? Like, basically, imagine you have a piano with 400 keys. No, seriously. You can strike a variety of different chords, but each key represents a set of neurons that carry that particular receptor. Okay? You want a key that plays A of the first octave to play A of the first octave no matter what. You don't want it to change, oh, A, but maybe C, but maybe something else. Okay, this is the signaling. You are somewhat familiar with this. And let's go over this slowly and steadily. So this is the odorant, and it binds to the receptor. Do you remember our conversation about a G protein? So this is your G protein, which gets activated and activates an enzyme, adenylate cyclase. This enzyme, its function is to convert ATP into cyclic AMP. Cyclic AMP opens the channel, cation channel, and sodium and calcium start diffusing into the cell. How are they charged? So when you bring positive charges into the cell, cell becomes depolarized, it releases neurotransmitter, you have a signal. Clear? Now it turns out olfactory neurons, the light yellow, these guys, they synapse with the next neuron called mitral cell in so-called glomeruli located within the olfactory bulb. So here's the question for you. You should be able to answer it. So imagine that, look at this glomerulus. This is a mitral cell that will carry the smell signal to the brain. These neurons that all together converge in a single mitral cell, do they all have different olfactory receptors or they have the same? And it's going to be cacophony. Because imagine this mitral cell will carry a bunch of signals. All cells that converge on a single mitral cell carry the same receptor. So this mitral cell will be, I don't know, vanilla. This will be cinnamon. And so on and so forth. Okay? Yeah, so all these receptors, all of them, neurons, carry same receptor. Now, another interesting thing about mitral cells. They have a high threshold of activation. What does that mean? What do you think? Again, not energy. It takes a lot of signal to actually trigger mitral cells, to deliver that, to produce action potential that will get to the brain. I have my own theory why that is so. We talked about senses before. Humans, when they were evolving, used to be scavengers. Okay? I'm terribly sorry I have to. So, humans used to be scavengers. If you eat dead bodies, you don't want your olfaction to be particularly sharp. Make sense? Yeah? Well, technically we're still eating dead bodies. Okay? Now I have a great opportunity to talk about, this is why I love olfaction so much. So, what I want you to understand about the pathways in the brain. Well, it goes, obviously, through the thalamus. Everything goes through the thalamus. Okay? But on its way to the thalamus, it activates amygdala and hippocampus. So if you remember the conversations about central nervous system, amygdala is one of the main structures in the memory formation, and it's responsible for the sensation of fear. There are smells that will alert you of a danger. That's your amygdala. Okay? Make sense? Did you have a situation when you smell something and it brings back vivid memories? Smell is a very strong stimulus for the formation of memories. I don't know if I told you about the smells of different states. Yes, but yeah, that brings different memories. Different smells bring different memories. Midwest smells pretty much the same. Yeah, like seriously, Kansas, Ohio, Indiana, Illinois. Now, why hypothalamus? Hypothalamus gives you physiological responses to smell. So imagine I kill 50 skunks and put them in the room, shut the door, and let them sit there for, let's say, 3 weeks. And then I open the door and let you in. What you gonna do? Immediately. Huh? You're gonna barf right away. This is the physiological response to a very, very bad smell. Okay? And I have a couple of great stories about the terrible smells. Does anyone have a gas stove? If you leave the gas open with no flames, I'm gonna smell it. The gas that burns has no smell at all. It's an additive called ethyl mercaptane, which gives the gas the particular odor. Ethyl mercaptane is a sulfur organic compound which is considered to be the smelliest and the most disgusting compound on earth. In sort of a more concentrated form, it smells like shit. Literally, like shit. Like feces. It's added in a very small proportion to the gas. Now, story number 1 happened in my alma mater, Moscow State University, 1 year before I started. So, ethyl mercaptane is very easy to synthesize, and a dude who was in OrChem lab decided to do it. And he did, in a test tube with a stopper. It's also very volatile, so the entire lab, like his classmates and his instructor, started to ask each other, why does it smell like shit? So he didn't want to get busted. He got out of the lab with a tube to the bathroom that is nearby. I know, like, I wasn't that, I did my OrChem labs in the same lab. I know the toilet. I know what we're talking about. So he made it to the toilet and realized that if he opens it, he's probably toast. So he opens the door, throws that tube into the bathroom, shuts the door, walks back. Obviously, tube breaks, but he doesn't care anymore. A few minutes later, another student, unsuspecting, goes to that bathroom, opens the door, immediately barfs. Like, there is no pause, there's no thought. Okay? To give an idea how all sulfur organic compounds are terrible. People who work with them, it's only partially a joke. When they ride a subway in Moscow, if there is a homeless person in the car, if the guy from Sulfur Organic Lab walks in, homeless people leave. It's too much for them, smell-wise. You cannot get the smell out of your hair or clothing. The only way to get it is to, like, shave your head and throw away the clothes. They usually have separate set of clothing. And there was a great story at Harvard University. There was a lab which worked on sulfur organics, and the administration, seriously, of Harvard, asked them to leave. They said, we're going to pay for relocation somewhere, but not on campus, because it was unbearable. So they were exploring nearby countryside and found a pig farm, like a big pig farm. Have you been to a big industrial pig farm inside? It smells really bad. It's really, like, cows are okay, pigs are disgusting. So they talk to the owner and say, you know, you have a barn, can we use it? And the owner says, yeah, sure, no problem. You're not going to surprise me with the smell. They move in, start working. 1 month later, the owner asks them to leave, saying that pigs are disgusting, but that's next level. Sulfur organics are terrible. Okay? Anosmia. Inability to sense smell. So several causes. If you go back to the anatomy here, sometimes people break their nose, and they can break cribriform plate. That will damage the filaments of olfactory nerve, and they will lose the sense of smell. Inflammation of nasal cavity. Now, if you're curious what causes the loss of sense of smell in COVID, nobody knows. Like, seriously. We know for sure that it's not the death of neurons. They are functional. But some kind of inflammation results in the loss of sense of smell. Huh? Yeah, but nobody knows why. Like, it's not your neurons that are being killed. It's something else. Olfactory auras. Important concept if you are working with people who suffer from epilepsy. Almost all of them, before they have an epileptic bout, have olfactory auras, which are short-term hallucinations, olfactory hallucinations. I had a student in my class who warned me that she has epilepsy and told me what to do in case of a bout. And I asked her, like, because I was freaking out. It's like, are you going to just drop from the chair? She goes, no. I'm going to have an aura. I'm going to tell you, and then you all can be prepared. Thankfully, never happened. No. It's now. Finally, taste. So, you taste with taste buds. Taste buds are located on your tongue in what's called papillae. You have 3 main types of papillae. When you stick your tongue out and see little dots on the anterior 2/3, those are fungiform papillae, and they're shaped like mushrooms. The folds on the sides of your tongue are filiform papillae, the foliate papillae. Folia is a leaf. And these, that's a poor choice of color. These are valate. Valate means there's a depression in the center. So this is the anatomy questions. Now within those papillae are taste buds. So within the taste bud, what you're going to find? You're going to find gustatory cells. So what I'm striking are gustatory cells. At the end of gustatory cells are so-called gustatory hair. Okay? And there are also the basal cells that can develop into a gustatory one. Can you kill those gustatory, the taste cells? Yeah, drinking something hot or eating something super spicy. Okay? Does that make sense? So the cells will replenish dead gustatory epithelial cells. Now this is the pore, and what fills up, well, not fills up, but what liquid is in your mouth? Why do you need saliva? Among other. In terms of taste, why do you need saliva? What does it do? Same. Dissolves the chemicals. Okay? If you can dissolve the chemical, you can taste it. Okay? We're going to talk about the formation of how sensation is formed. But I want to talk about different types of taste sensations. So sweet, well, everybody knows the sweet taste. I want to ask you, what is the evolutionary importance of a sweet taste from, like, for our ancestors 1 million years ago? Something tastes sweet. Why is that important? What kind of food tastes sweet? Food that contains a lot of which nutrients? Carbohydrates. Carbs. Easily digested carbs. Right? Sour. For our ancestors. Not poisons, but kind of rotten. Right? To be fair, it's now known that chimpanzees, did we talk about this? No? Chimpanzees seek fermented fruits to get drunk. Chimpanzees consume an equivalent of 2 to 3 beers a day in the form of fermented fruits. Okay? Salty. Why? Why did humans search for salty foods? Electrolyte. They wanted electrolytes. Does that make sense? Now, full disclosure, now in our diet, salt is not needed. We don't need to add salt. We receive enough sodium just with food. Salt is a spice now. Bitter. Why? What is the function? Okay. Excluding some types of beer, bitter taste. Would you eat something that is bitter? Huh? Probably not. So what does it warn you about? Yes, poisons. Poisons bitter. Okay? So, broadly speaking, alkaloids from plants are bitter. If you drink coffee, it tastes a little bitter, That's caffeine. Strong tea tastes bitter. That's theobromine. Have you ever tried like 99% chocolate? It's disgusting. It's theophylline, the main component in cocoa. Okay? And first person who yielded potassium cyanide in the lab, it was like late 1700s, tasted it, wrote in a diary it was bitter. He survived. He's fine. Was fine. He's dead because it was a long time ago, but yeah. And one of the reasons, if you read about like people being poisoned with potassium cyanide, usually the food that this compound is added to has to have very, very rich taste to mask the bitterness.
So it's not the same as when you were telling us it had to be spicy.
That's strychnine, but anything that will mask bitterness. Okay? Umami. What tastes like umami? Meat, cheese, any protein-rich food. There's an idea that maybe we need to include a sixth category, which will be something for fats. Okay? So, few myths. Taste buds have receptors for all taste modalities. Not that they're specific. Your tongue can sense taste, like any taste at any location. It's a myth that, like sweet is in front or like bitter is in the back. That's bullshit. Okay? And the mechanism. So, I will give you a very clear rundown. What you need to know about the mechanisms for the taste. So first, the mechanism for tasting the salt. Don't memorize those long scientific names. This is the sodium channel. This is saliva. When you eat something salty, there is a lot of sodium. Sodium diffuses along the gradient. That's it. How is sodium charged? Positive. Which means when it diffuses in the gustatory cell becomes depolarized. Does it make sense to you? Sour. Acid, as in sour, is hydrogen ions. So when you eat something sour, you deliver a fair amount of acid in your mouth, right? Two options. Hydrogen ions would use the same channel as sodium does. Okay? How are hydrogen ions charged? Positive charge, depolarization. Or they can open cation channels. See that? So cations like sodium and calcium will diffuse. Cations are positively charged. Depolarization again. Questions about the salt and the acid. All right, so I'm going to move on, trying to figure out which one is better. I think I want this image. So this image illustrates sweet, bitter, or umami. Now, hear me out. Do you need to memorize all the fancy names? No. Look. This is the receptor. Okay? This is the G protein. When G protein gets activated, it activates phospholipase C, the protein. Phospholipase produces this chemical called IP3, and this IP3 molecule opens the channel, allowing calcium to get out of endoplasmic reticulum. And that's sort of your final spot, because once calcium levels in the cell go up, it starts releasing neurotransmitter. So at the end of the day, 3 mechanisms. This one is for sweet, bitter, and umami. And these two are for salt and sour tastes, respectively. Does that make sense? Now, if we talk about the pathways, the only thing I want you to acknowledge, well, not only thing, the things that I want you to acknowledge. First of all, losing taste is hard. Why? You have 3 nerves, facial and glossopharyngeal, transmit taste sensations from your tongue, and vagus transmits the taste sensations from the pharynx and epiglottis. So you have a lot of taste buds all over the place, not just on the tongue, throat, lips, cheeks, epiglottis. Does that make sense? So even if one is screwed up, others will allow you to do the tasting. And the second thing, most of the what we consider taste anomalies are the smell anomalies. If you know someone who had the loss of anosmia due to COVID, I don't know, like I remember my conversations. People would say, well, coffee tastes just like a bitter black fluid, but it still tastes. It's just not the same, okay? To prove my point, I'm thinking about what we can do. I have an idea about couple of little experiments for you about sensations. But one thing that I suggest that you could do at home, I'm not forcing you. Can you tell the taste of sweet onion? Like if I will give you onions, sweet onions, will you be able to tell me that those are sweet onions?
You can tell you the onion, but I don't think it's sweet.
No, I mean there's different types, right? There's like what I'm saying is not like the biting, the spicy onion, just a sweet one. Yeah. I mean, I personally hate them, but you can try to do this. When you come home, ask somebody else to cut a piece of a sweet onion and a piece of regular apple. You can definitely tell those apart, can you? Then close your eyes and close your nose completely, just squeeze it. And ask the person to give you whatever they want. First and then second. You will not be able to tell apart a sweet onion and an apple. Moreover, if you will try to bite sweet onion with your nose closed and an apple with your nose closed, even texture is very similar. It's almost impossible to tell them apart because of the smell component. Okay.